You probably assume that every single nucleotide, like every letter in your DNA sequence, was just passed down directly from your parents, neatly packaged, locked away in the nucleus of your cells.
Right, like a secure biological vault.
Exactly. It's supposed to be your private vault. But when you look closely at evolutionary genomics, you realize the vault has actually been compromised.
Yeah.
Because throughout human history, this entirely different biological entity has been secretly copying and pasting its own code right into your private genome.
It really is wild. I mean, it completely upends that whole paradigm of a closed human genome. What we're actually looking at here is a phenomenon known as NUMTs, which stands for nuclear embedded mitochondrial DNA segments.
Yeah, and that is exactly the mission of our deep dive today. We are tearing into this massive new genetic study from Nature Communications. Now, we've known about NUMTs for a while, but historically they were just, dismissed as evolutionary junk or genomic fossils.
Yeah, totally ignored.
But this new study maps them across hundreds of diverse human and primate genomes and proves they are highly active. So, okay, let's unpack this. Take us back to the very beginning. How did mitochondrial DNA physically get into the nucleus in the first place?
Well, it all comes down to the endosymbiosis event. So mitochondria were originally independent bacteria that got absorbed by our cells billions of years ago, and they have their own distinct circular DNA.
Right.
But mitochondria naturally undergo a lot of turnover. They degrade inside the cell, and when they break down, their circular genomes degrade, and fragments of that DNA just kind of leak out into the cellular cytoplasm.
Okay, I like to picture this using an analogy. Imagine mitochondria as these ancient, really messy roommates who have lived in your cellular house for 1 billion years. And every so often, they just leave their personal belongings, their DNA, sitting out the communal living room, which is the nucleus.
Yeah.
And eventually it becomes part of the furniture.
That is actually a great way to think about it, because concurrently, the nucleus is always monitoring for double-strand breaks in its own DNA. And when a break happens, the cellular repair pathways, specifically one called non-homologous end, joining, they rush in to patch the gap.
But that pathway is, it's pretty sloppy, right?
It is highly opportunistic and error-prone. It just grabs whatever genetic material is floating nearby to bridge the break. So often, that material happens to be the leaked mitochondrial DNA from your messy roommate.
Wow, so the repair enzymes use the mitochondrial fragment as a literal patch, and it just gets stitched into the sequence.
Yes, and once it's in there, it's permanent. It gets inherited just like any other piece of your nuclear DNA.
That's incredible. But the detection aspect of this study is what really fascinates me. Because we basically missed the true scale of these insertions for decades, all because of the limitations of old technology.
Right, the short-read sequencing.
Yeah. If I understand correctly, traditional short-read tech basically chops your genome into these tiny 150 base pair fragments. And if a massive NUMT insertion exists in your DNA, but it isn't in the standard reference genome that the algorithm is checking against, the software just drops it.
It drops it, or it completely misplaces it. Because short-read algorithms rely on a linear mapping process. If it hits a complex structural variation, like a big chunk of mitochondrial DNA hiding in a nuclear chromosome, it just can't resolve the alignment against a flat, one-dimensional reference sequence.
So it's like trying to read a shredded document and missing whole pages, which is why the big breakthrough in this paper hinges on this new algorithm, the mini-graph cactus pangenome graph. Let's unpack that.
All right.
How exactly does a pangenome graph work?
So instead of comparing your DNA to one single reference human, which is like a single straight highway, the mini-graph cactus creates a multi-dimensional network. For this study, researchers used 538 haplotype resolved assemblies.
Meaning they looked at fully distinct maternal and paternal chromosomes from, 269 individuals?
Exactly. 269 individuals across diverse global populations. So when the algorithm sequences a read now, it doesn't just check one path. It evaluates these nodes and edges that represent every single known human insertion and deletion simultaneously.
And the data they got from this is crazy. By comparing against this pangenome graph, they achieved a 2.52-fold improvement in sensitivity. They found over 1,100 distinct NUMTs.
Yeah, 11,122 to be exact, and over 200 of those were completely newly discovered, just hidden in global populations.
And because the map is so robust now, they could take older data from 2,504 individuals in the 1,000 Genomes Project and genotype their NUMTs with near-perfect accuracy.
Yeah.
Like the Pearson's R correlation was 0.9998. that is essentially flawless.
It really is. The resolution is unprecedented.
Okay, let's pause and lock this in with a quick recap. Number one, NUMTs are mitochondrial DNA segments pasted into our nuclear DNA. Number two, old short-read tech missed them. And three, new pangenome graphs look at hundreds of diverse genomes at once, boosting detection by over 2.5 times. So, for you listening, think about your own genome for a second. If your DNA is this patchwork of ancient bacterial code, how much of what makes you you is actually borrowed from somewhere else?
Well, that leads perfectly into the genomic landscape of these insertions. The study categorized the NUMTs they found into 3 distinct groups. The first and largest group is fixed NUMTs. They found 774 of these, and they are present in at least 95% of all humans.
I actually have a mnemonic for this to help remember the traits of these fixed insertions. FIM. FIM.
Oh, I like that.
Yeah, so F is for fixed. I is for intergenic, meaning they land in the safe zones, the non-coding deserts between genes. And M is for mutated, because they've been there so long, they've accumulated a lot of mutations compared to the original mitochondrial DNA.
That's a great way to remember it. And that mutation aspect is crucial because it acts as an evolutionary clock. The nucleus has entirely different repair mechanisms than the mitochondria. So over millions of years, these fixed sequences accumulate specific C to T mutations, which lets geneticists calculate their exact age.
Right. And what about the other 2 categories?
So next we have polymorphic NUMTs. There are 280 of these, and they vary wildly among individuals. In fact, over 70% of them are rare or ultra-rare globally. And finally, there are 123 pericentromeric NUMTs.
Which means they hang out near the centromeres, right?
Exactly. Specifically enriched in acrocentric chromosomes where genetic recombination is just incredibly dynamic.
Yeah, wait. Here's where it gets really interesting. I have to push back a bit on the physical size of these things. The study notes that NUMTs can range from a tiny 31 base pairs to an absolutely massive 127,749 base pair mega-NUMT.
Yeah, made of concatenated fragments just stacked together.
Right. But shouldn't a massive 127,000 base pair chunk of foreign DNA be incredibly disruptive? Even if it lands in a safe intergenic region, chromatin has to fold into these precise 3D structures, these topologically associating domains or TADs, to fit inside the nucleus. Why isn't length the main factor for being weeded out by evolution?
It's a really intuitive question. You'd think a giant insertion would just destroy the spatial architecture of the nucleus. But the allele frequency data clearly shows that length does not negatively correlate with survival.
So a huge insertion is just as likely to survive as a small one.
Exactly. Because it turns out location matters way more than size. Chromatin architecture is remarkably flexible within those gene deserts. If a 100,000 base pair mega-NUMT lands in an empty TAD boundary, the genome just physically loops it out of the way.
Wow.
But if a tiny 30 base pair sequence lands inside a crucial protein coding gene, it breaks the gene and that lineage dies out. So fixed NUMTs survived because they landed in safe intergenic regions and avoided transposable elements.
Though the researchers did note that both fixed and polymorphic NUMTs love to hang out near segmental duplications.
Right, which act like biological copying machines. They essentially drive NUMT expansion across the chromosome without needing a fresh leak from the mitochondria.
Which perfectly sets up this fascinating anomaly the researchers found. When they looked at where the 774 fixed NUMTs came from in the original mitochondrial DNA, there is a massive bald spot.
That glaring absence, yeah.
A specific region called the 3' end of the D loop, or MTDL3, is almost completely missing in our nuclear genome. It spans coordinates 72 to 573.
Over millions of years, the nucleus actively rejected this specific piece of code.
But how did scientists figure out why? Did it just physically fail to integrate, or was it actively removed by evolution? What's the level of evidence here?
The evidence is really strong because they used an in vitro reporter assay. The researchers took human HEK293T cells in a lab, and they placed the MTDL3 sequence directly in front of a green fluorescent protein gene, or GFP.
Okay, so if the sequence does nothing, the cells stay dark. But if it acts as a cis-regulatory switch, like a promoter that turns genes on the cells, will glow green under a microscope.
And they lit up. This proved that MTDL3 has very strong intrinsic promoter activity.
Oh, wow. So if that sequence pastes itself near a random gene in early humans, it acts like a rogue electrical switch. It could turn that gene on permanently, causing chaos.
Exactly. Integrating it was likely highly disruptive, maybe causing early embryonic death or cancer, leading to its evolutionary deletion. What's fascinating here is that this shows the nucleus isn't just a passive trash can for foreign DNA. It actively filters out sequences that possess intrinsic properties that are incompatible with our biology.
Time for another quick synthesis.
Yeah.
1, we have fixed, which are common and safe, and polymorphic, which are rare and variable NUMTs.
Yeah.
2, size doesn't dictate survival, location does. And 3, the nucleus actively rejected the MTDL3 sequence because it acted like a rogue electrical switch.
Perfectly summarized.
So, audience, think about this. If our genome has been constantly editing out dangerous switches over millions of years, what dormant switches might still be hiding in your unique polymorphic NUMTs right now?
Well, the nucleus doesn't take chances with those new insertions. It actually uses a heavy chemical disguise to shut them down.
Because normally, mitochondria have basically zero DNA methylation, right? No chemical tags to turn genes off.
Right. But the researchers found that the second a NUMT enters the nucleus, the cellular machinery recognizes it as foreign and rapidly hypermethylates it. Over 90% of the sequence gets covered in these methyl tags, which tightly winds the DNA and effectively silences it to protect the genome.
But that silencing isn't always foolproof. Let's bring up a specific clinical case to ground this in human health. Among the 280 polymorphic NUMTs, they found a 42 base pair insertion that landed directly inside the coding sequence of the GTF2I gene.
Yeah, specifically in exon 25. And GTF2I encodes a really important transcription factor. It's actually located in the exact deletion region that causes Williams-Beuren syndrome.
Right, which is a severe neurodevelopmental disorder. So the mechanism here, the RNA sequencing data, showed this NUMT caused an in-frame insertion that created a premature termination codon. It basically dropped a stop sign right into the middle of the gene's RNA transcript.
Exactly. And that premature stop sign triggered a process called nonsense-mediated decay. The cell realized the RNA was broken and just shredded it. The gene function was completely destroyed.
Now, the carrier in the study didn't show the disease phenotype, probably because they had a healthy backup copy on their other chromosome. But it proves that these rare NUMTs can literally break essential genes.
They absolutely can. And it goes even further. Polymorphic NUMTs can act as subtle modulators of genes across the whole population. By looking at RNA-seq data from 731 individuals, they found 7 significant eQTLs, which affect gene expression levels, and 16 sQTLs, which affect how a gene is spliced.
So they basically dial the volume up or down on certain genes.
Precisely. For example, when specific NUMTs were present, the expression of a gene called GNL2, which is needed for nuclear export, went down. Same with SMD2, which regulates cell proliferation.
Wait, but didn't they also find one that increased expression? RASGRP3?
Yes, and that's critical because RASGRP3 is strongly linked to systemic lupus erythematosus and various cancers. The presence of the NUMT enhanced its expression.
So what does this all mean? It means NUMTs aren't just dead fossils. Your specific NUMT profile might be subtly tweaking your immune system or your cancer risk right this second.
They are active regulatory modulators.
Let's recap the clinical side. 1, the nucleus silences new NUMTs using heavy methylation. 2, rare NUMTs can physically break important genes like GTF2I via nonsense-mediated decay. And 3, some polymorphic NUMTs actively dial up or down crucial disease-linked genes like RASGRP3.
Exactly.
So knowing that a tiny 42 base pair insertion can completely terminate a gene's function, how do you feel about the fact that your genome is constantly at risk of being hacked by your own mitochondria?
It's a bit unsettling, but it's also the engine of evolution. If we pull back and look at the primate family tree, we can see this happening on a grand scale. The researchers compared human NUMTs to 20 high-quality, long-read primate genomes.
Right, our evolutionary cousins. And the insertion rates are wildly different, aren't they?
Vastly different. The pan lineage, so chimpanzees and bonobos, has a massive insertion rate. We're talking up to 18.7 new insertions per million years.
Wow. And what about gorillas?
Gorillas are much slower, only about 3.1 insertions per million years.
If we connect this to the bigger picture, why the huge difference?
It likely comes down to interspecies variations in reproductive strategies and cellular stress. If germline cells, the cells that make eggs and sperm, experience higher oxidative stress or longer periods of arrest, their mitochondria degrade faster. More degradation equals more leaked DNA getting pasted into the nucleus during meiosis.
Which provides the raw material for something totally wild the study discovered, a brand new mechanism of complex genetic variation. Variable number tandem repeats, or VNTRs, made entirely out of mitochondrial DNA.
This was a huge finding. We found a human-specific VNTR sitting in a gene desert on chromosome 12. In other primates, it's just a single NUMT, but in humans, it varies from 8 to 46 copies stacked back to back.
And the mechanism behind that is something called replication slippage, right? Happening at these AT microhomologies. Walk us through that. How does the copying machinery accidentally make 46 copies?
Well, when your DNA polymerase enzyme is copying the strand, it occasionally hits these repetitive AT sequences, adenines and thymines at the edge of the NUMT, and it literally slips. The machinery detaches for a microsecond, reattaches slightly backward, and just copies the whole NUMT segment again by accident. Over generations, this stutter expands massively.
They found another one of these stutters inside the intron of the MLPH gene, which is associated with human pigmentation and Griscelli syndrome type 3. In non-human primates, there are fewer than 7 copies, but in humans, it's expanded to up to 18 copies.
It completely reframes how we see these insertions. They aren't just baggage, they are the building blocks for massive human-specific genetic architecture.
It's just staggering. I mean, synthesizing all of this. NUMTs are active drivers of genetic diversity. They impact our disease susceptibility, our evolution, and they are continuously shaped by duplication and selective filtering.
We really couldn't see the true scale of it until these pangenome graphs allowed us to map the structural variations in multidimensional space.
So I want to leave you, the listener, with a final kind of mind-expanding thought. We tend to think of human evolution as something that happened in the distant past, right? Like it created this finished, polished product called the human genome. But if mitochondrial DNA is continuously leaking into your nucleus, creating new expression QTLs, dodging methylation, deleting regulatory switches, and expanding into these massive tandem repeats, your genome isn't a finished book.
Not even close.
It's a live open source document, and your mitochondria are actively writing the next chapter right now. So looking forward to the next million years of evolution, what will they paste in next?